Well, it's wonderful to be here, and actually it's quite a privilege to be the last speaker of this conference, especially in front of this audience who have known me for like 4 or 5 years with a lot of friends, colleagues, and mentors. Today I'm going to talk about a conceptual paradigm that we've been trying to develop in the lab since about 1.5 years, and hopefully by the end of it I've convinced you that deterministic processes can lead to stochastic outcomes. Right, so we study in Claude's lab, we study the classic genetic model organism, Drosophila melanogaster, to address fundamental questions in developmental neurobiology. And if you were to take a peek inside the head of this fly, you would see this structure. This comes from the EM connectome, which is the fly brain. And we study specifically a part of the fly brain responsible for visual system processing, and those are the optic lobes. And because we are developmental neurobiologists, we don't care about the function of this system, but how it grows and builds. So we study a more simple anatomical structure called the larval optic lobe. And what you see here are two populations of stem cells in pink and green. The neuroepithelial cells in pink are simply symmetrically proliferating and maintain the stem cell pool. But at some point, they get transformed into these green neural stem cells, which are the neurogenic population. And they divide to give rise to neurons. So how does this happen? Let's say you take one of these neural stem cells and look at what the behavior of that neural stem cell is. Something which is very unique about this system is that they undergo a transcriptional cascade, where transcription factors are expressed subsequently, one after the other, specifying the identity of the neurons which would be generated from that stem cell. So here you see transcription factor A coming before B and B before C, where the expression of the subsequent ones is driven by the previous one. And this is kind of a linear time series, and this would be a time series of one stem cell and each stem cell undergoing this cascade. But to produce neurons, you also need to divide. So what these stem cells also do is asymmetrically divide to give you two kinds of neurons because of an intermediate precursor population that occurs in the middle. So that's the general model that we've been studying for about 10, 15 years in Claude's lab. But what's not very evident in this diagram is that there are actually 2 times. And that's what I mean. So the first time is basically what I was talking to you about transcription factors, and it's called temporal patterning, patterning in time. And this changes the components of the stem cell to produce different neurons. And the other time is simply cell cycle. And so one of the things that we were trying to understand is how do these 2 times, or what is the relationship between these 2 times, where you have A, B, C, D, but you also have cell division 1, cell division 2, and cell division 3, and so on. So the main question of my project is, are these 2 time axes coupled? What is the relationship between them? And so if you were to look at the lineage in the beginning and assuming that it's completely coupled, you would get divisions at every stem cell, at every transcription factor window that the stem cell expresses. But if these 2 times were not coupled, you would get some instances where the cell cycle misses that window of gene expression, and you wouldn't get that cell type. So these are the 2 possibilities that I'm trying to distinguish. And for that, we use the neurogenesis in the optic lobe, which is quite a nice anatomical system to study this. So as I showed in the previous image, you have a lot of these stem cells at the surface of the brain, and they express the transcription factors one after the other. And as they keep expressing these transcription factors, they also divide and undergo cell divisions to give you the different neurons. And until the point you get this whole diversity of neurons which span the entire optic lobe. What we can do with the power of Drosophila genetics is there are about a few thousand cells here, a few thousand stem cells. We can label single stem cell lineages, like the clonal lineages of single stem cells, and ask, what is the proportion of these different neurons that are present within those clones? And if our first hypothesis was right, that they're completely coupled, this is what you would see, that each of these clonal lineages would produce each of those different neural types. But if the 2 times were not coupled, you would find gaps between them. And so this is the paradigm that I'm trying to distinguish, where is it deterministic or is it stochastic? So how do we study this? And let's get to the data now. We can make, using fly genetics, a lot of these different clones. And you see that is one of the clone as an example. But this is too dense to be analyzed. So you could take one of these different clones. And here you see about about 20 cells in a single clone. But because these clones are kind of induced randomly, you would also get different sizes of these clones. And simply by counting how many cells are present within a clone, you could infer whether it's from a single stem cell, double, or triple. And it kind of has a nice linear correlation of about 20 cells which are coming from a single stem cell. OK, so now I told you that these transcription factors are expressed in concentric rings, because that's how the anatomy is. So we have this one transcription factor called TFAP2, and it is supposed to label these rings of neurons that are born one after the other. And I'm just pseudocoloring these rings to show you that what we expect from this transcription factor labeling is to label 4 different cell types: TM1, TM4 in the earlier ring, TM2 and TM6 in the later ring. But as you can see right now, we don't know what is the relationship between these cells and the stem cells that they were born from. So what we need to do is basically make clones along the diagonal and ask how many of those clones contain these different cell types. Well, that's what I did. And when you make those clones along the diagonal, the first observation that was extremely surprising to us was that the null hypothesis is to expect 4 in a single clone. But most of the time, we ended up finding only 1 in each ring, so that is 2 in the clones. And we think that's because of the variability in the system. But what's even more surprising is that when we push the system to make more clones along this axis, we find sometimes clones with 2 in the first ring and none in the second ring. And it kind of points to this stochasticity which might be occurring in the system, where you cannot, with complete precision, predict how many cells you would get whenever a neural stem cell divides to produce different cell types. And here we're focusing mainly on neurons. And this is just an example where we're showing 4 cell types. That was here. But I've done this analysis for a couple of other cell types. And you can see that there is quite a lot of variability. And so in this plot, the null hypothesis was that if you would see neurons being produced from each stem cell without any stochasticity, it's completely deterministic, all of these clones represented by the dots would be at the 1 line. But you actually see quite a lot of variability. But some cell types don't show that variability, interestingly. And that's something that we're going to follow up later on. In the end, I hope I've convinced you with this data so far that there are 2 deterministic timing mechanisms, 2 clocks in some sense, right? Temporal patterning and cell cycle. And they're both quite deterministic because temporal patterning goes A, B, C, D in terms of transcription factors. And cell cycle goes G1S, G2M, G1S, G2M. But when they come together in a single stem cell, the result of these deterministic processes can be stochastic. And that's what we're studying. For future directions, we would, well, we have, but I haven't presented this data because it's still preliminary, manipulate cell cycle and ask how does that affect the proportions of cell types which are born from one stem cell. And also, how does cell death play into controlling this variability? Because a lot of this variability would also be not really healthy for the fly. And with that, I would like to thank our labs in Abu Dhabi and New York. Well, firstly, my mentor, Claude, who's been quite amazing as a mentor, and the CGSB organizing committee, Stefan, Chris, Inas, the labs in Abu Dhabi, and also CGSB members, Khalid, Paula, Susan especially, the New York lab, especially Yen, my thesis committee members, and Ken, who's here. Thank you for coming, and thank you all of you for the attention.